Slideshow. Okay. All right, so we've learned about hydrolysis and dehydration synthesis of polymers. So again, all living things, humans, microbes, and everything in between are comprised of 4 major macromolecules of life. You understand the term macromolecules? What does macro mean? Big. So big molecules. These are biopolymers of life. Carbohydrates, lipids, proteins, nucleic acids. Okay? Got me? Okay. So carbohydrates. We'll talk about those first. So carbohydrates. Here are 2 famous ones, glucose and fructose. Are they isomers? Yes, they are. They have the same chemical formula, different shapes, yeah? One of them they have OH groups, alcohol groups. Do they make you drunk? They only make you drunk after they've been converted into ethanol by yeast, yeah? So sugar does not make you drunk, right? If you have a 2-liter bottle of soda and you pound it right before you drive home, well, that's bad for you, but it's not going to make you crash your car like alcohol will, yeah? Even though it has OH groups all over it, yeah? In the same way that ethanol does. Go figure, right? The functional groups change the properties of the molecule, but the way the molecule behaves inside biological systems can be hard to predict, okay? All right. So carbohydrates, we know of them as an energy source, right? Glucose is grape sugar. It's an energy source. We burn it for energy. There are other uses for it, but that's a major use. Likewise with fructose, fruit sugar, that we find in an apple or an orange. Okay, so the general formation of glucose is into a ring structure. Again, these 3 pictures are all the same thing, glucose. They're in slightly different conformations. This is the linear conformation, which does exist in nature. The 2 on the right are much more common. One is a simplification of the other. You can see each carbon atom in the picture on the left has a number, right? That is because glucose has a particular shape, and it cyclizes in a particular and predictable way, and that's here. And this shape can be simplified as the drawing on the right. Again, the drawing on the right doesn't even bother to show you the carbon atoms. It shows you the oxygen that's part of the ring because that's unusual, okay? Is that clear? All right. So again, glucose molecules are modular, yeah? We can assemble them and disassemble them, right? One carbohydrate we call a monosaccharide. So glucose is a monosaccharide. Mono means 1, saccharide means sugar, yeah? So 2 monosaccharides linked together by what process? Dehydration synthesis forms a disaccharide, a dimer, yeah? Does that make sense? That is maltose. Do you guys know maltose? Do you know the taste of malt? You ever have Whoppers at the movie theater? Raise your hand if you had Whoppers at the movie theater, right? They have a sort of a different taste, a malty taste. Yeah, it's a little different. It's a little less sweet. So that's, again, there. Sucrose is table sugar. This is regular sugar we have in our sugar bowl at our houses, right? So sucrose has the molecular formula C12H22O11. When we take glucose and fructose and assemble them by dehydration synthesis, that's what we get. So, are we surprised by this chemical formula? You should look at it a second, and maybe you shouldn't be surprised after that second, but maybe think about it a second. What is the chemical formula for glucose? Glucose, C6H12O6. Fructose, what's the chemical formula for that? You can find it on the same page. It's an isomer, so if you know it's an isomer, you know the chemical formula is the same, C6H12O6. And when we assemble them together, we should have C12, H24, O12, but that's not what we have, do we? We have C12H22, O11. Everybody please briefly consider this. And then if you have an explanation, please raise your hand. And don't give away the answer. Shh. Let everybody think about it for a second and have the pleasure of solving this problem, or the sting of not solving it and hearing the answer. So I see some hands up, and that's good. Good on you. Has everyone had a chance to think about it and try to solve it? Okay, raise your hand for an answer if you want to spat out an answer. All right, Kelsang, what do you got? They share covalent bonds, right? So when they come together to share covalent bonds, why is this not C12H24O12? Why is it C12H22O11? Because the water came off from what? From dehydration synthesis. So here it is. This is from your textbook, yeah? There's the water. You guys understand? C6H12O6, C6H12, C12H22O11 plus H2O gives you C12H24O12. Does that make sense? Got it? Okay. And again, if you thought about it, got it wrong, and felt a little sting, that's a good sign. That means you're going to remember it next time, yeah? Yeah. So there, it sounds like your question is, why did glucose and fructose form sucrose instead of something else? So why does glucose and fructose form sucrose, whereas glucose and glucose forms maltose? Is that the question? Well, I think the answer to that is because we are starting with isomers, not the same molecule, right? And so these molecules as glucose and fructose are already different. If we have two of the same molecules stuck together, then we have that. If we have two slightly different molecules stuck together, we have that. And as far as the shape, you can see that glucose is a sort of a hexagonal shape, whereas fructose is a pentagon, yeah? And the pentagon, one thing that's different about it is each of the hexagons has a carbon coming off the top, yeah? So there's 1, 2, 3, 4, 5, 6 carbons in glucose, but there's also 6 carbons in fructose, yeah? 1, 2, 3, 4, 5, 6. Does that make sense? And the reason for that is because the fifth, if we count down these carbons, 1, 2, 3, 4, 5, this one links with this oxygen. This one links with this oxygen. Does that make sense? And so for each, there's an extra carbon on the bottom, but for this one, there's also an extra carbon off the top. Does that make sense? But this carbon is one closer to this, so the ring is a pentagon instead of a hexagon. All right, is that satisfactory to you? So think it through a little longer, and if you have more questions, come back and ask me again. So we have other sugars, right? Lactose is made out of galactose, which is a hexagon, and glucose, which is another hexagon. We do have 5 carbon sugars. This is deoxyribose and ribose. Have you heard of these? Where do we hear about these? DNA and RNA. Hopefully you guys know that. If you don't, you'll find out soon enough. Don't worry. So we've made sugar monomers, glucose and fructose, into dimers, disaccharides, sucrose, maltose, and lactose, yeah? But again, there's no limit to the length of the chains we can make. If we take glucose and instead of linking it together to form maltose, we can keep going and adding maltose after glucose after glucose, and we can have chains that are thousands of glucoses long. And when we do, we have 3 different kinds of arrangements in which we can have those molecules. So these are all glucose monomers that form starch, yeah? Understand? These are all glucose molecules, and we find starch in potatoes, yeah? We can find glycogen in our muscles. How many people have heard of muscle glycogen? If you exercise, you do endurance sports, they always talk about muscle glycogen and liver glycogen and yada, yada, yada. So it's kind of like starch, only it has branches off of it. And because animals need to be faster than plants because we can run and they can't, then we can, anytime our muscles are working, we can break a glucose off and burn it to get energy to exercise, or we can break it off here, 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 and here, and here. We can access the energy a lot faster by breaking the glucoses off one by one off of many branch points instead of just 2. Does that make sense? Likewise, if the fibers are arranged in a slightly different way with what are called beta bonds instead of alpha bonds holding the glucoses together, then the fibers, instead of forming spirals, form straight fibers that hydrogen bond with each other, and those fibers are cellulose. This is wood or cotton or pretty much fiber from your food like celery. Or spinach or something. So cellulose is a structural molecule that plants can make using the glucose that they make from photosynthesis. Okay? Guys with me? All right, so you guys understand that these are polymers. These are theoretically infinite molecules. How long is a starch molecule? Well, it's usually thousands of glucoses long, but it could always be 1,001. It could always be 500. Does that make sense? Likewise with cellulose. They are theoretically limitless polymers. Right? And again, bonds and branching differentiate the major polysaccharides, starch, cellulose, glycogen. Why do we talk about this? Well, because bacteria can form these things also. Bacteria can make glucose polymers, and then they can take those glucose polymers and cover themselves with them. This is called a capsule. And a capsule is a structure that a bacteria can form around itself, and it can make this bacteria harder for our bodies to kill. Right? So these are actually pictures of strep. And these strep bacteria have capsules around them, and because they can have capsules, these kinds of strep are nastier and harder to kill than bacteria that don't make these capsules. And these capsules are basically made out of glucose polymers that make their cells slimy and sticky and gooey and insulated. Okay? Is that clear? So bacteria that we learn about in class can defend themselves with carbohydrate polymers covering their bodies. Okay? Furthermore, here's glucose monomers can be modified with functional groups. So here's a special glucose polymer. You see the glucoses all in a chain? The glucoses are not quite glucoses. They are modified glucoses. So take a glucose. This isn't glucose quite. This is N-acetyl glucosamine. Have you heard of glucosamine? Anyone take glucosamine pills for their knees? If you've got rickety knees, sometimes people will take glucosamine. What do you think glucosamine is? It's glucose with an amine group on it, an amino group, yeah? So on this amino group, we can put an acetyl group. That's another functional group. So this is a fancy functional group that modifies every single one of those glucoses, yeah? So take a bunch of modified glucoses and string them together, and you've got chitin. Chitin is what bug shells are made out of. It's what mushrooms are made out of. If you're ordering pizza today, which I will be doing, then the mushrooms, you will be eating glucosamine in the cell walls of the mushrooms on your pizza. Yeah. Also, if you ever heard of those stitches? You get stitches and then the stitches sort of disappear, right? Your body can digest glucosamine, right? So or it can digest chitin, right? So if you make your stitches out of chitin, then the enzymes in your body will gradually break down those stitches, hopefully not before your skin heals. Does that make sense? Okay. So, lastly, a very important modified glucose polymer. Do you understand what I mean by modified glucose polymer? Yeah? Modified glucose. That means glucose with a functional groups on it, extra functional groups that regular glucose doesn't have. Yeah? Polymer. You understand that term too, yeah? All right, so this is a modified glucose polymer. Here is the way it is. This is peptidoglycan. Bacterial cell walls in nearly all of the bacteria we'll be discussing in this class, both gram-positives and gram-negatives, make peptidoglycan. Peptidoglycan is a very important biological polymer for microbiology class, okay? Is that clear? All right, so here it is. A bunch of NAGs and NAMs alternating, right? What the heck is a NAG? That's N-acetylglucosamine, the same thing that was in chitin. We don't get an N-acetylglucosamine. We get an N-acetylglucosamine with a muramic acid on it. What the heck is muramic acid? I don't know, this thing. Yeah? You understand? It is a glucose monomer with some modifications to it. Yeah? You get your car from the car dealership and then you take it to the post-manufacturing modification, you get your fancy wheels. Yeah? These are your fancy wheels on your glucose car. Yeah? Does that make sense? Okay. So, N-acetylglucosamine, N-acetylmuramic acid, right? NAG-NAM, 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 over and over and over and over and over. That is peptidoglycan. Or that's the base part of peptidoglycan. Okay? Is that clear? And why do we care about peptidoglycan? Because it forms the cell wall of pretty much all bacteria. Why do we care about a glycocalyx? That's outside the cell wall. That is a outer shell outside the cell wall that some bacteria have and others don't. And the ones that do can be nasty pathogens. Okay? Is that clear? All right. Sucrose can be hydrolyzed into, boom, D is correct. Nicely done. All right. You guys getting tired? So it's about lunchtime. You guys want to take lunch? Okay, so here's the deal. Here, I want to stop this first. Yes.